Welcome to this video on muscles. This video will introduce foundational concepts for understanding the histological structures and functions of skeletal, cardiac, and smooth muscles. I hope you enjoy it. Muscles are tissues that contract either voluntarily or involuntarily. Their functions include contractility, movement, stature, and the control of blood pressure. There are three kinds of muscle in the human body. The first is skeletal muscle. This is a voluntary muscle that is usually attached to bones for gross movements of the human body. The second type is cardiac muscle. This is an involuntary muscle that makes up the heart. The third is smooth muscle. This is also an involuntary muscle that is located in many tubular organs throughout the body, one example of which is the gastrointestinal tract. Let's begin by looking at an overview of skeletal muscle. Before we look at some examples of muscle, I want to introduce you to spaghetti logic for understanding the two sections that you will often see muscle in under the microscopes. Muscle cells are long cells, and so you will often encounter them in longitudinal sections. And this is akin to looking at spaghetti lengthwise. In this analogy, each piece of spaghetti corresponds to one muscle cell, and I'll show you more of what I mean by this on the next slide. Likewise, if you now turn the spaghetti on its end, this is akin to what muscle cells look like in transverse or cross sections, again with each piece of spaghetti corresponding to one muscle cell. Let's now use that longitudinal example of spaghetti and compare it to how skeletal muscle looks in longitudinal section under the microscope. Just as I said, each piece of spaghetti corresponds to the appearance of each individual skeletal muscle cell, three of which I've just labeled. Take note that the white spaces between muscle cells are artifacts of tissue preparation. The key feature of skeletal muscle cells is that each cell has multiple nuclei, and the nuclei are eccentrically placed to the periphery of the cells. The reason that they have multiple nuclei is because skeletal muscle cells develop embryologically through the fusion of multiple myoblasts. The reason that the nuclei are pushed off to the periphery of the cells is because the cytoplasms are packed full of the contractile proteins actin and myosin. If we continue with a longitudinal section of skeletal muscle and find a good section, we can see another defining characteristic of skeletal muscle, striations. In this image, the muscle cells are running horizontally, and we aren't seeing any white artifactual spaces in between the cells. What you are seeing, if you compare it to this schematic of parts of two muscle cells, are striations crossing the muscle cells, as shown by the dark pink lines in this schematic. Are you able to see the striations in the microscopic image on the right? Aren't they beautiful? These striations are caused by the organization of the contractile proteins actin and myosin within the cytoplasm. They are best understood at the electron microscope level. If you are interested in learning more about this, then a quick Google search will satisfy your curiosity. Turning the spaghetti and muscle in a different direction, let's now look at how skeletal muscle looks in cross-section. Let's start off at a fairly low magnification to show you how muscle cells are organized into larger structures called muscle fascicles. When you think of one of your skeletal muscles in its entirety, you are thinking of multiple muscle fascicles all bound together by connective tissues that are given special names in the muscle of epimysium, endomysium, and perimysium. Let's look at one of these muscle fascicles in cross-section at higher magnification. If we magnify one fascicle, then we can see the individual muscle cells with their eosinophilic cytoplasms and their basophilic nuclei. Now compare the microscopic image to a schematic, and you can identify the multiple eccentrically placed nuclei that are characteristic of skeletal muscle cells. You will not see this organization of nuclei in the other two muscle types, cardiac and smooth muscles. Let's now take a look at cardiac muscle, which is involuntary and is located in the heart. Let's start with a longitudinal section through cardiac muscle. Here I have labeled 3 muscle cells. You can see that, just like skeletal muscle, they are long cells. However, unlike skeletal muscle, the nuclei are not pushed off to the side, and you can see that reflected in the rather plump, round shape of the nuclei of these cardiac muscle cells. Just as skeletal muscle has unique features, there are 2 unique identification features for cardiac muscle. The first is that the cells branch. I'm showing that here schematically on the left, with the yellow line traveling down one cell and branching into 2. Now watch as the yellow arrows on the microscopic image on the right travel down one cell and branch into 2. The second unique feature of cardiac muscle is the presence of intercalated discs. If we start by looking once again at a schematic of cardiac cells, this time 2 adjacent cells, then I am now adding in a dark pink line in between the cells and indicating it with an arrow. This is an intercalated disc. They can also nicely be seen on this microscopic image here on the right, with the dark pink arrows each pointing at an intercalated disc. Intercalated discs are only found in cardiac muscle cells, and they are involved in ensuring that the heart is beating as a synchronous unit. Again, if you are interested in learning more about the cell biology of intercalated discs, a quick Google search will satisfy your curiosity. Finally, let's have a look at the appearance of cardiac muscle in cross-section. Unlike skeletal muscle, which is composed of large cells with multiple nuclei around the periphery of the cells, cardiac muscle cells are comparatively small, and they have only one nucleus that is very centrally located in the cell. You can see this by comparing a schematic of a round cell with a single centrally located nucleus to the microscopic image of cardiac muscle cells with single centrally located nuclei. This brings us to the final muscle type, smooth muscle, which is involuntary and is located in many tubular organs, one example of which is the gastrointestinal tract. Smooth muscle is very much like cardiac muscle in terms of having one nucleus that is centrally located in each cell. However, what sets smooth muscle apart from cardiac muscle is that it can be very difficult to distinguish individual smooth muscle cells in both cross-section and longitudinal section. The cells all seem to blend into each other, and all that you will see is a lot of pink cytoplasmic material and a lot of basophilic nuclei. You will not routinely see the cell borders of smooth muscle like you did with skeletal and cardiac muscles. For those of you who like such things, here is a comparative summary of the key features of the 3 different muscles that I have introduced today. The cross-section images are on the left, the longitudinal images on the right, and in the middle is a summary of the key features of each muscle type. I hope it's helpful. This concludes the introductory video on muscles. Thank you for watching. You may access the virtual slides used in this video by clicking on the UBC Virtual Histo link in the banner of this YouTube channel.